Well, good morning, good afternoon, or good evening, ladies and gentlemen. Welcome to another installment of our video PowerPoint series for Unit 6, DNA, RNA, and Protein Synthesis. Today, what we're going to be talking about primarily is DNA and how it replicates. What we're also going to be talking about towards the end are specific types of mutation that can occur either at a certain point in the DNA or that affect the entire chromosome itself. So, without further ado, let's go ahead and get this thing started. I will see you on the next slide. So, DNA replication. Simply put, replication is the process of copying a DNA molecule. So the genetic information can be passed on from one cell to another. Now, DNA, if you think back to mitosis and meiosis, DNA always replicates before cells divide, during the S, or synthesis phase. So basically, this whole process is just making more DNA or doubling it, so when we go ahead and split the cell, we have an equal amount on both sides. So a quick review. DNA is made up of two strands of nucleotides. The strands are held together by something called hydrogen bonds that are between each of the bases. And the two strands are complementary with each other, meaning that A, or adenine, is always going to pair up with T, or thymine, and C is always going to pair up with G, so cytosine is always going to pair up with guanine. Just a quick review here. Now, the hydrogen bonds that hold them together are extremely important, but we'll talk about that a little bit later in the show. So how this all works. During replication, each strand is used as a pattern, or a template to make two new DNA molecules. This is called semi-conservative replication, because what we're doing is we're taking the DNA, splitting it in half, and then basically filling in the sides that are missing. So each new strand contains half the old material and half the new material. That's why we call it semi-conservative. So we have our original strand, and what we do is basically using an enzyme called DNA Helicase, we go ahead and split it up into two separate halves. Now, as you can see, we got some bases that need pairing. So what we do is we basically go through and add in the missing bases. So now we have the original left strand with the right new strand, and the original right strand with the new left strand. So as you can see from this diagram here, we've got half the old material, and we've got half new material. So that's why we call this semi-conservative. So the steps in DNA replication. Step number 1 is the double helix unwinds. So we go from this nice twisted ladder formation to more or less a just straight ladder. Then using the enzyme DNA Helicase, you can tell it's an enzyme because remember it ends with ASE, the DNA Helicase unzips the DNA, breaking the hydrogen bonds between the strands. Now this is important because, this is where we were coming back to why we need hydrogen bonds. Hydrogen bonds, if you think back to the biochem unit, hydrogen bonds are very weak. Now this is important because we need these bonds between the strands to be weak so we can break them relatively easily. If we had to go ahead and waste all this energy breaking the DNA, we'd have to waste so much more of our energy going ahead and replicating our cells. So, basically what happens is we have our DNA strand, and then we meet DNA Helicase, which as you can see, goes through and unzips the DNA into two separate strands. So we have our DNA Helicase, which is our enzyme, and we have the unpaired bases on each side. Step number 3, another enzyme by the name of DNA Polymerase, pairs up the free nucleotides in the nucleus with nucleotides on the DNA strand, forming new hydrogen bonds. So inside the nucleus, we actually have free-floating nucleotides. So we have free-floating adenine, thymine, guanine, cytosine. And what the DNA polymerase does here is it goes through and matches everything up. It's like, okay, we're missing an adenine here. So we have an adenine, no pair. Let's put a thymine with that. So where do these nucleotides come from? Believe it or not, they come from what you eat. So as you go ahead and ingest food, that energy is also being used to create more nucleotides. Now that does not mean if you're like, oh, well, I'm feeling a little deficient in adenine, I'm going to go ahead and eat some eat some avocado or some apples. You know, I'm feeling a little deficient in cytosine. I'm gonna go ahead and eat some carrots, because you know that starts with C. Sorry, that's not how it works. So, steps in DNA replication. We have our original DNA, which is unzipped by what? Take some time. So the DNA is unzipped by DNA Helicase. Then using DNA Polymerase, we go through and put in the free nucleotides that have been floating around inside the nucleus with the unpaired bases on the opposite strand. So the free nucleotides pair up with the original left strand and the free nucleotides pair up with the original right, giving us two completely new strands of DNA. So the DNA then, once we have everything paired up, winds back up into a double helix. The final products of DNA replication are two molecules of DNA, each having half new material and half old material. This is why we call it semi-conservative. So, we go ahead, have the DNA, unzip it, pair up the other sides that need it, we rezip both DNA molecules, and bingo, bango, bongo, we got ourselves two new molecules of DNA. So, like we said before, here is where we started to go ahead and split the DNA using DNA Helicase. So we split it up into two separate strands. Then, using DNA Polymerase is going through and adding in the missing bases to both sides, going ahead and creating two new strands. So, why do we need to replicate? Why should we? So, we need to replicate because now all the genetic information can be passed on to the new cells during cell division. So we start off with our original cell. We copy the wow. That was beautiful. So we go ahead, copy the DNA, so now we have twice the amount. We need twice the amount, so when the cell splits, we have two identical cells that now each have a copy of the original DNA. So if you'd like to, there's a video down, I think it's going to be down here, or maybe down here depending on where I'm at, but it shows you a nice introduction to how replication works and why we need it. So if you want to go ahead and take a look at that, I'd highly suggest it, and I will see you all on the next slide when you come back. Seriously, check it out. Now the other thing I'd like to talk about real quick is something called PCR, or polymerase chain reaction. If you want to go ahead and venture into the world of forensics, or investigations, or anything like that, this is something that you will be using constantly. So what we can do here is we can actually make millions of copies of DNA from a very small sample. So, I don't want to go ahead and spoil about how it works, but seriously, check this video out. It's awesome. And kind of terrifying at the same time, of how quickly we can go ahead and replicate these things. So, same as before, check it out, and I'll see you when you come back. So, accuracy. Replication is incredibly accurate. Only 1 in a billion base pairs are incorrectly paired. Now, when a mistake is made, the DNA molecule can normally correct itself, so the genetic code remains the same and nothing gets hurt. However, sometimes they can't, sometimes they miss it. Even the most diligent of eyes sometimes miss something. And these are what we call mutations. Oh yeah. So gene mutations. We're going to talk about two different types of mutations today. We're going to talk about gene mutations, which occur inside the DNA, and we're going to talk about chromosomal mutations, which occur inside the chromosome, or the supercoiled DNA. Give me a sec here. Cup of coffee in the big time. Yeah. So point mutations, or gene mutations, happen at single nucleotide points in the DNA. That we can have things like substitution. So with substitution, one nitrogen base is exchanged for another. So you kind of sub one out for the other. So we originally had an adenine, and we replaced it with a guanine. So we're swapping out one for the other. We can have an insertion. So with an insertion, this means that a base gets put in. So if we originally had AAT, AAT down the DNA, we would now have something like AGAT. As you can see, one got inserted into the genetic code. We can also have deletion. So with deletion, a base is removed. So if you had AAT, now it would just be AT. We lost a base. So the important thing to remember here is that this is all happening at one nucleotide. Just one. So, the dog ate the fat cat. Why would I be telling you this? Well, there's a reason. Check this out. The dog ate the fat rat. Now think back. We talked about 3 types of mutations that can occur: substitution, insertion, and deletion. So the dog ate the fat rat. What's it look like we did? Well, if you go ahead, look at the original. It originally said the dog ate the fat cat, and we replaced the letter C with the letter R. That would be a substitution. Now, what does this even mean? The oga tetif ater at. What? So if you look at it again, look at the original up top, and then compare it to the one down below. And you might be able to see what happened. Go ahead, take your time. So if you look down here, we're missing something. What happened is we actually removed the D. So when we got rid of the D here, what are you noticing? So the D was originally right here. So when we deleted that letter, everything shifted one letter to the left. So by going ahead and deleting that, we caused a pretty big mutation, and that's deletion. Our final one here, the dog tet eth effa trati. Once again, that makes no sense at all. But if you look a little bit deeper, you might be able to figure out what happened. So if you look down at the T here, there was no T before. So, same thing, but instead of shifting everything to the left like we did with deletion, we shifted everything one down to the right. That's what happens with insertion. So as you can see here, out of these 3 mutations, which one seems to cause the least amount of damage? Which one makes the sentence still something you can roughly understand? That'd be substitution. Substitution isn't really that bad of a mutation. But if you have deletion or insertion, as you can see, it changes the whole sentence, taking it from something that originally made sense to something that you can hardly understand. And the same thing happens with your genes, with your DNA. So, what do we have here? What kind of point mutation do we have? We originally had CTG, GAG. But now we have CTG, GGG. That would be a substitution. Bingo. Let's take a look at this top one first. So, we originally had AUG, CGA, UUA, UAC, GGG. And with our second after the mutation, the first one's still the same. No problems there. AUG, AUG. Next we have CGA, CGA. AUA, AUA. But if you look here on our fourth one, there's an A now that wasn't there before. And sure enough, everything got shifted down to the right, one letter. That means we have an insertion. Now if we look down here at the bottom, AUG, AUG, hey, everything's good. CGA, CGA, yeah. UUA, UUA, we're in good shape. But here, what happened? The C has actually been deleted. So when that happened, everything shifted one letter down to the left. That's what we call a deletion. So insertions and deletions can cause something called frame shifts inside the genetic code. So a frame shift, simply put, is just any time we have a series of bases shifting down to the left or to the right. So like we said before, one nucleotide affected every codon, which is a series of 3 bases, which eventually will code for our amino acids, which make our proteins, one single shift caused a change in everything. So instead of going ahead and creating what our body might potentially need, it's not getting that. This is why frame shifts can be so potentially dangerous. So what we're going to talk about now are chromosomal mutations. So we talked about genetic, gene, or point mutations. Now we need to talk about chromosomal. So remember, the point mutations happen only at one nucleotide. The chromosome affects the entire chromosome, which are thousands and thousands of molecules of DNA. So, what seems to be happening here? If you look up top, A, B, C, D, E, F, G, H, I, J. When at the bottom we have A, B, D, E, F, G, H, I, J. Looks like our C went ahead and got deleted. So deletion, same thing as with a point mutation. Part of the chromosome is just simply removed. For our second type here, we originally had A, B, C, D, E, F, G. Now we have A, B, C, D, E, D, E, F, G. Then in our third one, we have A, B, C, D, E, D, E, D, E, D, E, F, G. So what seems to be happening here is we have a duplication. So a duplication is where one or more segments, in this case, the D and E, are replicated or repeated time and time again. They're duplicated more than once. That's what we call duplication. Now here, A, B, C, D, E, F, G, H, I, J. And in the bottom, we have A, B, C, D, E, F, I, H, G, J. So if you go ahead and look at this closely, it's the same letters, but now they're all backwards. So what happens here is we get something called an inversion. With an inversion, a stretch of DNA is reversed within the chromosome. So what happens is we basically get it flipped around completely. So instead of being GHI, we now have IHG. So an inversion is any time we have a series of the chromosome that is flipped backwards. Now here we have something a bit different. So at the top, A, B, C, D, E, F, G, H, I, J on one chromosome. Then on our second chromosome, we have K, L, M, N. Now what seems to be happening here is after the mutation, we have A, B, C, D, E, F, G, N. And then for the other chromosome, we have K, L, M, H, I, J. So what happened here is one part from one chromosome was swapped with one from another. Now these are not homologous, meaning they are not for the same trait, which can cause some damage. We call these translocation. So with translocation, entire gene segments are exchanged between non-homologous chromosomes. So let's say hypothetically this top one was for, let's go blue eyes. And our second one here, the KLMN, was for blonde hair. Now sure enough, we have a blue coloring on the hair and a blonde coloring on the eyes. Could cause a pretty groovy mutation, don't you think? Now, once again, that's just a simplified version of how this could work, but that's translocation in a nutshell. Let's keep going. So, a little bit of a checkpoint here. I want you at home to go ahead and figure out what type of mutation all of these are. I can give you a hint, they're all chromosomal. So go ahead, take your time. I'll be sitting here. I already know what they are. I want you to figure it out. Pause the video if you'd like. Alright, I'm going to give you a little bit more time. Alright, that's enough time. So, with our first one, what seems to happen here? Looks like the B was taken out. So this would have to be our deletion. In our second one, we go from A, B, C, D, E, F to A, B, B, C, D, E, F. You better believe it, what we have right here is a duplication. For our third one, ABCDEF. Sure enough, now look at this. We have AEDCBF. So we look at our original strand and our mutated strand. We have the same these same sections of chromosomes, but they are inversed. So they're backwards. So that would be inversion. And finally, the only mutation we know of that involves two different chromosomes that are non-homologous would be translocation. So we went ahead and exchanged two parts of two different chromosomes that resulted in two new ones. That's translocation, all right. Now, mutations. Let's talk about mutations for a sec. There are three categories of mutations. Mutations can be silent or neutral, so they have no effect. You don't even know that they happened. Now, believe it or not, most mutations are silent. So most mutations don't cause any damage. You can't really see any effect they had. The second type is harmful or fatal. They have a bad effect. Some mutations can be incredibly damaging. You know, if they occur. Let's say there was one that was for how your body developed, and there was a deletion there causing you to not grow a certain part of your body, or that it would be hard for you to go ahead and replicate something. That could be pretty bad. Now the final type, now these are very low probability here, that the mutation could be beneficial, so it could have a good effect. Now this is a very low probability. For example, if you look down here at the scorpion actually has two tails, twice the stinging power. So that would be a pretty good piece of good news for the scorpion. However, the fact that beneficial mutations can occur is incredibly rare. Very low probability. Well, ladies and gentlemen, thank you for joining us today for DNA replication and mutations. So, in summary, what we talked about today was how DNA, we go from one strand to two. We also talked about how different mutations can arise. We talked about gene mutations or point mutations, which occur at only one spot. For example, insertion, deletion, or substitution. Substitution being the least damaging of the three because we're not causing a frame shift. Insertion and deletion cause the entire code to shift either one down or one up, causing way more damage. We also talked about chromosomal mutations. So with chromosomal mutations, we talked about insertion. Sorry, not insertion, that was a different one. We talked about deletion. So part of the chromosome is actually removed. We talked about duplication, where part of the chromosome is repeated over and over. We also talked about inversion, where we take part of the chromosome and flip it backwards. And we also discussed translocation, which is when we have crossing over between two non-homologous chromosomes. So two chromosomes for different traits that swap information. Well, once again, I'd like to thank you for joining us today, and until next time, I will see you in the next video. You all keep it classy.